Hello, and welcome to this CooperSurgical Fertility and Genomics Solutions seminar. My name is Tony Gordon. I'm the Vice President of Business Development here at Cooper Genomics, and I'll be giving a short introduction to PGT-A and our technology here at Cooper Genomics. And then I'll be following on to introduce Professor Darren Griffin from the University of Kent, and Darren will talk about some of the interesting latest research and some comments on the PGT field. So today we're going to be predominantly talking about PGT-A. That's pre-implantation genetic testing for aneuploidy, testing chromosome biopsies from embryos to see if they have the right numbers of chromosomes. It's controversial. Does it improve success rates? And Darren will talk about some of those controversies following this. Is it an option for patients? So that's what we're going to talk about here. We're not going to talk about PGT-M, which used to be known as PGD, and that's looking for finding embryos that don't harbour a disease, so we don't pass it on to next generations, or PGT-SR, looking for typically chromosomal rearrangements in embryos and trying to avoid those unbalanced embryos. So we're going to talk about PGT-A. So PGT-A is really quite simple in theory, at least. So the theory is if you can find an embryo with the correct number of chromosomes, 46 chromosomes, then you can implant that embryo in preference to other embryos, and it should end up with a higher likelihood of pregnancy. It's controversial, but that's the theory, simple in premise. Here you can see an embryo biopsy, and we've schematically shown that it has 47 chromosomes here, and there are 3 copies of chromosome 10. So if you go to a cytogenetics textbook and look up trisomy 10, you'll see there's never been a live human being born with trisomy 10 in all their cells. So we know that that embryo wouldn't make it, so we deselect that embryo and select other embryos in preference. So this is the premise. So you try to avoid those embryos with aneuploidy, and it's a major determinant of fertility, which increases with maternal age, so a major problem. And if you do implant embryos with aneuploidy, you might get failed implantation, miscarriage, or even genetic disease like trisomy 21 Down syndrome. So here you can see a pretty well-known graph. Down at the bottom we have increasing maternal age, and in blue you can see aneuploidy increasing with maternal age. So most of that aneuploidy is coming from the egg, and most of it is determined by maternal age. And as aneuploidy goes up, of course, the number of euploids, the number of embryos with the right numbers of chromosomes, goes down. Hence it's harder to find embryos, and hence with and without PGT-A, it's harder to get pregnant with advanced maternal age. Although if you can find those euploid embryos, a euploid embryo is fairly independent of age per embryo transfer in terms of success. The other complication that we have are mosaic embryos, and these are events after fertilization where you get mistakes in replication of chromosomes, and some cells in the embryos will be euploid and some cells will be aneuploid. And that's just an added complication to the work that we do. Here you can see an example of 2 mosaic embryos. The one on the left is a mixture of euploid and aneuploid, and it's a mixture throughout the embryo. So not just the inner cell mass, but also the trophectoderm. Whereas the one on the right is aneuploid across the trophectoderm and the inner cell mass. And we're learning more about embryos all the time. There was a really interesting presentation at ASRM earlier this year, which Cooper Genomics contributed some of the data to, and that was from Zuva's Fertility, and they looked at the implantation of 1,000 mosaic embryos. And what we can say is mosaic embryos implant less, they miscarry more, but they can sometimes lead to healthy live births. And really what people are doing are selecting euploids first, and then if there's no euploids to select, they might consider mosaic embryos for transfer. So here at Cooper Genomics we have this 4-way measure of reporting embryos now. Euploid, and aneuploid, and then about 14% of embryos are in this low-level, high-level mosaic group. And you would consider transferring the euploids first, and then you might consider the low-level mosaics next, and then the high-level mosaics. And the highest mosaicism score determines the score of the embryo in our system. So these are the results from 75,000 embryos that we've just collected recently with our latest PGT-A algorithm. And you can see here that we're doing very well from less than 1% of embryos with no amplification, what we call no intact DNA, and 2% of embryos where we couldn't get results because the biopsy was too noisy or whatever. Then just under half are euploid, 44%, so they're suitable for transfer, which is really good given a 37-year maternal age average. And then we have low-level mosaics and high-level mosaics. And if you put these together, about 58% of embryos could be considered for transfer. So it's still an important group here in the mosaic embryos that we can consider for transfer. So the things that we've been doing over the last few years is taking our technology, which is DNA sequencing to count chromosomes, trying to improve that technology. And we did that first with our first iteration, which was called PGT-AI. So instead of taking a baseline from cell culture cells, we actually built a baseline, built an algorithm on the outcome of real data from biopsied embryos that had resulted in a live birth or sustained pregnancy. So that became our real world baseline. And then we validated that using 10,000 embryos, which was done manually and done by the algorithm. So what it does, it takes away looking at these profiles by human interpretation, and it becomes statistical and just called on p-values, and it avoids subjective interpretation. It also makes sure that there's no errors in transcription that you get sometimes when you get people doing this sort of analysis. We've gone on from that to our 2.0 version of the platform, and that was possible because we were able to go from smaller sequences to larger sequences and use the power of those larger DNA sequences. So we were able to get 10 times more data per sample. We're still using the same baselining technology, but we resequenced all those samples using a more accurate sequencing method, giving us the ability with that 10 times more data not to only count chromosomes, but also count what's known as the SNPs within chromosomes. So you have two methods now for looking for any chromosomal changes. The standard sequencing DNA counting, and also the SNP counting of chromosomes, which is more linked to inheritance rather than numbers of chromosomes. So our standard 2.0 test uses these two methods, chromosome counting and SNPs. That removes artifacts. That's how come we get such a low no result rate now, low QC fail result. It gives us 5 megabase resolution, which is very good to see smaller things, and it also gives us best in class mosaicism calling, which is really interesting now that we see that 14% of samples with mosaicism. What we can do then as well is we can unlock more data from that, open the door to more data by taking the DNA from the maternal and paternal providers of the gametes. And that's really important because there's a bunch more things we can do. So we can still do the same tests, but we can do additional things now. We can look for the origin of aneuploidy, which is really useful if somebody gets the results back and every embryo is aneuploid. They might want to know the origin of that aneuploidy to make decisions about things like gamete donation or continuing with their infertility journey. It also allows us to detect all forms of triploidy or haploidy. That's where there's either 69 chromosomes in the biopsies or 23. So where you have 46 and it's become 69, that's triploidy. instead of 46, you have 23 haploidy. That's important because they have implications to ongoing pregnancy. Also, we can identify whether the 2PN check for FER check was correct as well, and that is becoming increasingly interesting to try and get the data from every embryo that's cultured. So we've advanced very nicely with our new 2.0 platform. It enables us to do our two tests together on one sample, copy numbers and SNPs. It gives us more data, allows us to see smaller changes, whether that's in size or in mosaicism. It detects all forms of ploidy when we're in those parental samples. It adds in parent of origin testing and 2PN validation. If you go to the link here on our YouTube channel, you'll be able to find more about PGT-A 2.0, some of the webinars we've given recently, and some of the other information we have on that. So PGT-A, that's where our technology is. Some things are fairly clear that it does. So if you can find a euploid, almost regardless of the age until the early 40s, that embryo will have a similar chance as a younger maternal age embryo, and that's fairly well described. Also in the papers and in the clinical trials, it reduces miscarriage rate compared to not using PGT-A, and it increases implantation rates. Things that are more controversial is whether it increases live birth per cycle started, so whether it increases live birth per what's known as intention to treat or per treatment cycle. Also, whether it reduces time to pregnancy is open to debate, but it does enable more confident single embryo transfer, and I think the data is now becoming clear that with PGT-A we see more single embryo transfer. So just in summary, the PGT-A AI technology aims to improve IVF outcomes. It advances the PGT technology to give you a more complete understanding of the embryo genomic content, whether that is through the copy number analysis or with the SNPs or the two together. And it gives hopefully more choices and potentially more outcomes for the patients by being able to give as much information as possible prior to a transfer. So that's where we're going with the PGT-AI 2.0 technology. So that's a little introduction as to where we are, and now I'm going to pass over to my colleague, Professor Darren Griffin from the University of Kent. Darren has been in the PGT field since it really started. He was the first person to do gender for medically indicated requirements using FISH, and he's been key to a number of the advances in the PGT field over the years, such like the introduction of carrier mapping, which is the PGT-M technology that our lab and many of the labs across the world work on. So Darren's going to talk a little bit about PGT and some of the more controversial things that are going on in our field at the minute. So without further ado, over to you, Darren.
Okay, well, first of all, welcome to the cabin at the end of my garden. I'm very grateful for the opportunity to speak to you all, and thanks to all at Cooper for giving me the opportunity to do so. My name's Darren Griffin. I'm Professor of Genetics at the University of Kent, and I'm going to tell you a little bit today about whether PGT-A works, and in particular, some analysis of the HFEA data. So if I start with conflict of interest, I do receive research funding from Cooper, and thanks very much again for you sponsoring me to speak here, but also from other companies as well, such as competing companies such as iGenomics. I don't have any shares in any companies anywhere. I did once share a room with Santiago Monet and threatened to shoot him, but that's a long story. But really, I don't have any conflict of interest. So if we think about PGT-A, then we have a number of referral categories. There's advanced maternal age, recurrent pregnancy loss, recurrent implantation failure, male factor infertility, And a number of people have suggested that it should be used for all IVF cycles. So I'm going to structure my talk today thus. First of all, how do we interpret the data, including the RCTs? Secondly, mosaicism. Thirdly, are there any deleterious effects of doing embryo biopsy? And also just going to give a little thought to stubborn people and how they might be impeding the whole process in general terms. So let's do the first of those. How do we go about interpreting the data, including the RCTs? Now, if we think about the evidence base now, and this is abstracts, published papers that I've seen over the years, I think I've counted up to between 60 and 70 retrospective studies involving array CGH or NGS or both that report a beneficial effect of PGT-A. So something like you see an implantation rate with PGT-A is higher than regular IVF, particularly for the older patients. So can we please stop talking about fish studies? They are no longer relevant anymore. I saw a Cochrane review recently that largely concluded that PGT-A was not a good thing because they'd put together all the randomized trials, including the fish studies. And I just don't think that's a fair and scientific way of doing things. There are some RCTs involving array CGH and NGS, however, about 7 in total. There is a non-selection trial, which I'll talk about. I'm going to tell you a little bit about some new data in cattle. I'll talk a bit about some multi-center analysis that we've been involved in from Care Clinic. I'll mention the SART data as well, but also predominantly talk about some recent analysis we've done on HFEA data. Now, I got this slide from Santiago Bonet and I adapted it. Now, I can tell you that when he presents it, all the bottom row here is in green. He would tell you that it's all positive and everything to do with RCTs in PGT-A is positive. I don't think that's true. I think each of these studies, although most of them, has some positive and some negative. And so, for instance, some of the early ones were criticized because of the small sample size. And as well, you know, the STAR trial showed no significant overall improvement, but nonetheless, an OPA per transfer significantly improved for the older age groups. So there are some positives and some negatives in each, or at least most of these studies. These are the results from David Scott's group on the non-selection trial. And if you can draw your attention to the right-hand side of the graph here, you'll see that they have a sustained implantation delivery rate of 0% where an embryo that they have diagnosed and called as aneuploid is implanted, compared to a 65% chance if they've diagnosed it as euploid, which is really very compelling and highly significant. I would also like to present some data to you from my lab. This is an analysis of some cattle data in which cattle embryos were generated, they were biopsied, they were all subject to a SNP chip, but for the purposes of looking at estimated breeding values. One of the things that we did was we went back and looked at that data for aneuploidy. And I can tell you that when we diagnosed normal embryos, so euploid embryos, the pregnancy rate was nearly 60%, and the live birth rate 46.5%. However, if we diagnose the embryo as abnormal as a result of the biopsy, then the pregnancy rate and the live birth rate were down to about 4%, so very few got through. So this is really quite similar to the non-selection trial of David Scott's group that I just showed you a moment ago. Now, equally, if we take that same data, identical data, and I have to tell you that this is highly statistically significant, if we looked at it in a different way and said, well, overall, with this 50% abnormality rate, how much does it improve the pregnancy rate? Then the answer is 6.2%, and the live birth rate, 5%. And this is only just statistically significant in comparison. So one of the things, so this is a little bit like the randomized control trial. So one of the things I would say to you in all earnestness is please do look objectively at your data. If you look at it one way, it could be highly significant. In another way, it might be a little bit different. There's nothing magic about statistics. Now, as many of us will be aware, the SART data, the pregnancy outcome data from the US, tells us that the PGT-A versus controls is relatively level. So in the lower age group, under 35, we don't see much of a difference whether we put PGT-A in or not. However, if we look at the older age groups above 35, then we do see a significant difference. And this difference was reflected in the older age group of the STAR randomized controlled trial, in which you can see this data is almost identical, in which there is a significant improvement of, what would that be, around 15% or so, 13, 40% maybe, in comparison. I'd like to present to you some new data. So this is from Catherine Sanders in my lab, who was supported by Cooper. And in this case, we've looked at, in purple, the live birth rate per embryo transfer in when we used array CGH, when we've used NGS in blue, and then in black for non-PGT-A. So, and then in this case, you can see that there are really some differences. Now, if we superimpose whether this is significant or not, if it's, so the numbers are the actual numbers of transfers, the gray means that it's not significant, the yellow means that it is significant. And again, I think we can see a number of things here. First of all, other than in clinic D, so I should say that A, B, C, and D are individual clinics in the care fertility group, all using the same SOPs, of course, but only clinic D saw a significant difference. However, only saw a significant difference in the younger age group. All of the others, by and large, with one or two exceptions, showed a significant improvement, sometimes using array CGH, sometimes using NGS, and usually using both in these older age groups. Now, obviously, for instance, the right-hand side of clinic B there, up here, didn't have very many numbers to be able to make that call, only 3 in the array CGH and none in the NGS. So some of them are not significant because of lack of numbers, but by and large, you can see that there are highly significant differences. And then what I'd like to present to you is the results that we got from the HFEA. So on the 13th of July, 2020, we submitted a Freedom of Information request to the HFEA, in which we asked for their data for 3 years, from 2016 to 2018, on live birth rate per embryo transferred, live birth per treatment cycle, number of embryos transferred, and the percentage of cycles where no embryo was available for transfer. There were around 190,000 cycles that we looked at. I think it was 190,010, of which nearly 2,500 were PGT-A. And again, this is the work of Catherine Sanders in my lab. These are the results. So in every age group, for live birth rate per embryo transfer, the PGT-A was significantly greater than the non-PGT-A, and more markedly so the older the patients got. Similarly, the live birth rate per treatment cycle was significantly greater in the PGT-A group compared to the non-PGT-A group, again, even in the under 35s, but again, more markedly so the further we got in maternal age. When we asked the question of what proportion of cycles were there where no embryo was transferred, then you can see the under 35s up to, well, really the under 40s are not dissimilar, but thereafter we start to see a difference, such that for the over 44s, we did not find in half the cases an embryo available for transfer, which is about half that in the IVF. So you can interpret this in a number of ways. You can say, well, these poor patients didn't get the opportunity at all. Or you could say, we perhaps identified those embryos that would have never implanted or lead to a live birth anyway, so we save them the pain of an embryo transfer. When we consider the mean number of embryos used per transfer, you can see that for PGT-A, it's almost always 1, rarely anything else. Whereas multiple embryo transfers, 2 or more, is really quite commonplace in the non-PGT-A. Now, we all know that there is an issue of obstetric complications with more than 1 embryo transfer. So one interpretation of this data is that by and large, PGT-A is allowing the transfer of just single embryos on most occasions. So the HFEA data tells us this, that up until 2018 at least, 1.3% of all IVF cases currently in the UK are PGT-A. All the live birth differences in all the graphs were statistically significant, even the under 35s. And this tells an actually quite similar story to the SART data, the age breakdown of the STAR trial and the non-selection trial. So it's confirmatory evidence. It tells us that it facilitates single embryo transfer in nearly all cases, unlike regular IVF, which has health issues, particularly for older women. And it also identifies instances where no embryo is suitable for transfer. So how do we interpret this evidence base? Well, by and large, single center cohort retrospective studies largely point to higher implantation rates and live birth rates following PGT-A, especially in the advanced maternal age category. The multi-center analyses point to efficacy in the over 35s, the CARE data, the new analysis, the SART, and the HFEA new analysis also in the younger age group as well. The non-selection trial, the NST, provides strong evidence that aneuploid embryos are unlikely to develop into live birth, and we reflected that in the CATAL studies. Admittedly, the RCTs are somewhat mixed, but most display some positive benefit for PGT-A. So one of the things I would entreat the community to consider is the whole of the evidence base. You're talking about evidence-based medicine. That doesn't just mean RCTs, it means everything, in my view. So we always think, and particularly in reproductive medicine, think of the balance of the evidence. And in this case, my reading of the situation is this appears to favor PGT-A. Which then leads me to the thorny issue of mosaicism. So let's start with a definition. Mosaicism is the incidence of 2 or more chromosomally distinct populations in an embryo or fetus, and this includes the placenta. So in the whole of the conceptus, you've got at least 2 different populations of chromosomally distinct populations of cells. What it is not, however, is the detection of normal and aneuploid cells in a 5 to 10 cell trophectoderm biopsy. If you observe that, unless you've got something wrong in your analysis, which can happen, but the NGS results traces I think are pretty clear. If you observe that, at 20% to a 1 out of 5 abnormal trisomy, then you almost certainly have a mosaic embryo. By definition, you have, because you've got at least 1 cell which is chromosomally different from the rest. But if you don't observe it, if you have all your 5 cells are normal, or all of your 5 cells are abnormal, then I would argue that the data suggest that you still probably have a mosaic embryo. And thus, a trophectoderm biopsy is only a rough measure of the level of mosaicism. So it will tell you that you have some degree of mosaicism. You probably have it anyway, but don't sort of take it as read, right, we definitely got 20% there, we definitely got 40%. So if I were to review all of the literature for you in terms of mosaicism in human embryos, then it would take me a couple of hours. So that's for another day. And it's fair to say that all the studies say something slightly different. However, you can certainly interpret the data reasonably convincingly to say that all embryos have some form of mosaicism. And we know that a lot of mosaic diagnoses can lead to normal live births. So this is a table from a paper that a book we have out at the moment, clinical outcome after transfer of mosaic embryos. So for the reasons I've just given, I've crossed out of mosaic embryos to read following mosaic diagnosis of biopsy. Okay, because I think they're all embryos. I was only personally involved in the last of these studies, but you can see that when mosaic diagnosis is returned, you can still get reasonably good pregnancy rates. Now, mosaicism isn't just a uniform thing. There are many, many different ways in which mosaicism can arise. And nonetheless, you can break it down roughly into three. And the first I call chaotic, that we know that a small minority of embryos have 5 to 10%, say roughly, have multiple abnormalities with a different abnormality in every cell. Okay, so that is definitely mosaicism. And well, these are not going to proceed to term. So surely we should be eliminating these by PGT-A. If there's an initial error in meiosis, and this could be 2 to 20% of embryos in younger women, up to 50% in older women, maybe even more, this can lead to a pathology. It almost certainly does. It is related to maternal age. Some have questioned whether it's related to ovarian stimulation, but again, we rarely lead to unaffected conceptuses. These have pathologies. So surely, again, we should be eliminating these by PGT-A. Let me just go into this in a little bit more detail. So imagine that we have a meiotic error leading to, in this case, an oocyte with 24 chromosomes, a zygote with 47, but then some sort of second error event or a rescue event, embryo correction, it's sometimes called, such that a proportion of the cells are normal again. We know that this does happen. Then you will get mosaicism, definitely. It's a well-described mechanism, but I think it's fair to say that more than 50% of cells will be affected. But just imagine that we took a biopsy here. We would say, well, 3 out of the 5 cells are normal. So this is a low-level mosaic. But if we took it here, we would say they're all abnormal. So we need to be a little bit careful about making global interpretations of the overall level when a blastocyst at this stage only has a couple of hundred cells. But the one that we're most worried about is the post-zygotic mosaics, because this could be the remainder of all the embryos, and these are trickier. Now, the incidences do vary between clinics, and it does make you wonder whether there's something to do with the culture conditions, so that's something certainly to look at. But whether they have a pathology or not, then depends on a number of things. So it depends on the proportion of cells that are affected. It depends on the germ cell layer that's affected. It depends on the chromosome involved. I think this is one of the areas in which research is sadly lacking, and we need more of it. So in this case, we start off with a normal zygote with 46 chromosomes, some sort of post-zygotic error that could be independent chromosome loss or gain, or mitotic non-disjunction. And by and large, this is more likely to be less than 50% cells affected. So for instance, if we took a biopsy here, we might see 5 out of 5 cells. If we took it here, we might see 1 out of 5. But if we took it here, it might be 5 out of 5 normal. So it is a little bit difficult. Again, let's not just imagine that our interpretation of the overall level is necessarily accurate, but nonetheless, the presence of mosaicism in the biopsy probably is. Now, mosaicism is not something that was discovered as a result of looking at IVF embryos. We've known about it for a long, long time. It causes intrauterine growth retardation, intrauterine death, placental insufficiency, uniparental disomy, high and low birth weights, and stillbirth. And in the studies back in the 1990s, doing regular karyotyping on just 2 germ cell layers of the chorionic villus, come to the conclusion that probably around 30% of all trisomy conceptuses are mosaic. And what this graph shows you, why it's so boxy looking, is that they've looked at the extraembryonic mesoderm of the chorionic villus and also the trophoblast, which is derived from the trophectoderm. And it basically tells you that it's usually confined mosaicism. And in point of fact, the more you look for it, the more you'll find. So this 30% is an underestimate. But there are also very chromosome-specific patterns. Chromosome 2 and 3, for instance, behave very differently. But importantly here, in the context of PGT-A, is if we look at this graph a little bit more closely, there are black dots and white dots. And the black dots means there is a pathology. The white dots mean that there isn't a pathology. Okay, so most of the time with a mosaic, it proceeds normally to term. If it's an error of meiotic origin in the first instance, then we usually get a pathology. Very rarely do we not. But more than half of our post-zygotic trisomies are clinically uneventful. And we know, in fact, that at least 2% of all normal pregnancies have post-zygotic trisomy cells. And again, the more you look, the more you'll find. So this 2% is probably, again, somewhat of an underestimate. Which leads us to the question of when we consider the array CGH versus the NGS. Now, undoubtedly, the NGS is a better technology. It's a better dynamic range. You get a cleaner trace. It is much, much, much better. But if you are detecting these mosaics in which you've just got an odd cell, or maybe 2 cells out of your 5, are you statistically just picking up more of these low-level mosaics that would be clinically uneventful? So although we've improved the technology, and we've heard from Tony about some artificial intelligence as well, although we've improved the technology for detection, and the AI does that even more, we do need to also revise our interpretation as well. Which leads me to my third point. If we're biopsying the embryo, is this affecting it in some way? Now, I did ask a colleague of mine whether they were ever going to put together a list of tips and tricks of doing embryo biopsy. Now, I've never biopsied an embryo in my life, but when I read a list like this, and I don't expect you to read every word, I don't expect you to agree with absolutely everything that's written on there. It is nonetheless a very, very long list. And this tells me that unless you do it right, you can detriment the benefit of the development of the embryo. So it really is simple. Of course, embryo biopsy can damage the embryo if you do it badly. It can compromise embryo development, but it also can provide to the labs a poor starting material, which makes it more likely to return an unclear result. So studies that do it badly will not show a benefit of PGT-A, particularly the RCTs. Just because a study is well designed, an RCT does not necessarily be a given that it's well performed. But if you do it well and follow the guidelines, such as the ones I just showed you, then it should not damage the embryo and it should work. And this really reinforces the need for proper external quality assessment. So the people who are very, very good at this, to get it out there and to properly train in all these subtle little tricks and tips to do it well, efficiently and quickly, so it's back in the incubator as soon as possible. Which then leads me to my fourth point. Now, there are a number of points of view, shall we say, about PGT-A, and I'm expressing in a sort of straw man way an extreme one. So some people would say there is sufficient evidence completely and absolutely to justify the continuation of PGT-A. Well, we just can't wait for those RCTs because the clinics just wouldn't survive. We depend on the employment of our staff. We need to innovate quickly. and the innovation is good. And it's all right saying, you know, we're just fleecing patients, whatever, but look at that clinic down the road. They don't innovate. They just wait for trials all the time, and they've got very low success rates. We'd have never had IVF if been subject to the prior rigors of an RCT, nor would we have ever had ICSI. And look, when these RCTs are designed, they take years, they're poorly funded, so your numbers aren't necessarily quite so high. We might be pretty convinced already that the benefits are obvious, and the appetite to the trial may have waned halfway through it being performed. Now, equally, on the other side, the argument is that no, absolutely not. PGT-A is not properly validated until there are proper RCTs with intent to treat criteria. And we must do this. And the reason is that clinics are motivated by the need to be seen to be innovative and making money, despite the proper supporting evidence. These add-on treatments are just not good, generally. Any treatment not validated by an ICT should only ever be part of a trial. It's unethical, it's unfair to patients to subject them to these treatments unless the case is proved with at least one RCT. How do you sleep at night if you don't believe this? Well, those are two extreme views. And in a talk this afternoon, I will be going into the HFEA guidance and what it's recommended for PGT-A. And we'll park that for now. But what I would have hoped to think in a group of talented, free-thinking people is that if we had empirical evidence to support PGT-A, of course we'd all have different points of view, but you know, it would be roughly bell-shaped, and we would shift a little bit more towards the left if it was against PGT-A, and a little bit more towards the right if it was supporting PGT-A. Now, this is not an empirical study, this is just what I think I see, but it seems to be that every time we get a bit of empirical evidence, all that happens is the majority of people dig in their heels. So whether it's positive or negative, it just seems to polarize opinion. It's like talking about Brexit sometimes. But I think there's an awful lot of things that we can agree on, and this should be our starting point. So we can all agree that we don't do cleavage stage biopsy anymore. There are some labs that can claim with evidence that they're good at it, and some trials have shown a benefit. But when we roll this out into the real world, it seems that it makes the embryo too prone to damage. So let's just park that. Let's just say we don't do cleavage stage biopsy anymore. We don't do FISH anymore. There are better methods out there. Some people can argue that it might be effective, but we don't do it. So why are we talking about it? Why is there a Cochrane review about it? We can all agree that PGT-A will not improve cumulative pregnancy rate. So if that's your one and only measure, then fine. But if we are also interested in pregnancy rate per cycle, in time to first pregnancy, and miscarriage rate, that could, and the evidence suggests that it does, be effective in PGT-A if it's performed correctly. And I think we can also agree as well that there is not yet sufficient evidence to justify it being routinely rolled out in all IVF cases. Now, I think that the vast majority of people, I haven't done the study, but I would guess 90 to 95, maybe even 99% of people would agree with all of these things, no matter how pro or anti-PGT-A they are. So let's do that as a start, and let's stop. Let's take all these other things like cleavage stage biopsy, FISH, cumulative pregnancy rate off the table. We all agree that. So then let's return to our referral categories. Advanced maternal age, recurrent pregnancy loss, recurrent implantation failure, male factor infertility, and also the question of all IVF cycles. Does PGT-A improve IVF success rate in advanced maternal age? Now, I would argue the retrospective analyses, the large data set analyses, the non-selection trials, the cattle data that I showed you, the RCTs to some extent, and the HFEA's own data, on balance, point to it being effective and safe. But only for time to first pregnancy, pregnancy rate per cycle, and chance of miscarriage. not cumulative pregnancy rate. There are, I think it's fair to say, some concerns about the RCTs, and yes, we do want more. Okay, we should never be entirely contented and happy. But I would say that on balance, there is only one direction that the evidence is pointing in. Now, should we use it in the referral category of recurrent implantation failure? Well, possibly. But RIF is a very complex issue. There are endometrial issues as well. And I think it's fair to say that even well-designed, well-performed trials may not show an effect given all these other complications. But given the non-selection trial, if we were doing it, trying to treat recurrent implantation failure, do we really want to be transferring embryos that have little or no chance of live birth? Do we not want to get that? If we're going to say we're going to do a program of treatment for you, do we really not want to include that? And I would argue that possibly we do. For recurrent pregnancy loss, again, this is very complex. Advanced maternal age is easily defined. When's your date of birth? Recurrent implantation failure and recurrent pregnancy loss are not easily defined. Recurrent pregnancy loss is also very complicated, and even a well-designed, well-performed trial may not show an effect unless the patient group is stratified in a particular way. But it may provide peace of mind if someone had a previous pregnancy loss that was aneuploid. Male infertility, I think at this stage, we just don't know. It's the least common referral category anyway, and either way, we need more evidence. Routine IVF, I think we can safely say not yet. But given the NST results, we need to keep looking, and it may become a prospect sometime in the future. So this is where I think we are at the moment, but we can improve. Of course we can. Because I don't think the current categorization, euploid, aneuploid, mosaic embryo, is sufficient. I think it really, really does the whole field a disservice. I think, given what I've said about mosaicism, chaotics and meiotics and so on, that really we should have a bit of a rethink. Based on the diagnosis that we see, okay, we should be getting rid of the chaotics, not transferring those, they're not going to work. We should be getting rid of anything that shows a meiotic abnormality. We can do that now with sequence-based data or SNP-based data. We shouldn't transfer trisomy 21s, 18s, or 13s because of the risk of effective live births. And indeed, monosomies or deletions can lead to pathologies. So let's just get rid of those. Don't transfer them. Anything in which an abnormality is not detected, then quite possibly we can consider that to transfer. So no abnormality detected, not euploid embryo. The embryo is mosaic, I can tell you. But if we see something that may be compatible with a normal live birth, such as a post-zygotic mosaic trisomy, in which you maybe seen 1, 2, 3 cells, some segmental duplications that may be also mosaic, then this is where those lovely people that we know as genetic counselors come into their own, because they can consider the balance of the evidence, the individual circumstances of the patient, the level of the abnormality, the chromosome involved, how do they feel about biopsy and analysis, what other embryos are out there, and then to perhaps stratify the embryos that might be abnormal. And I think this is a far more sensible approach than this very binary, if you like, normal, abnormal, or maybe mosaic. So how do we make PGT-A better? I think we have to have a balanced interpretation of the whole evidence base, including the RCTs, not just imagine the RCTs are the be-all and end-all. The HFEA data is a very good example. Mosaicism is not a simplistic issue, and we shouldn't just go, it's mosaic, okay? We need some intelligent interpretation along the lines of the traffic lights that I said. Is there any deleterious effect of biopsy? The evidence from a number of groups suggests no, as long as you do it right. Okay, so if you do it right in an individual lab, if you want to roll it out across the country or around the world, then proper EQA is needed. We've got to stop being stubborn people. We've got to stop using the evidence as confirmation bias of our own prejudices. We have got to take a look at the evidence and be prepared to modify our view. Be more appreciative that other people have a different point of view to what we have. And we need to embrace new innovations. Tony spoke about AI. We might get something an awful lot better out of nanopore sequences, for instance, and spent medium analysis. Will that deliver on its initial promises? We need to be open and receptive to it, not just dig in our heels and say it won't work, or indeed say, oh, this is wonderful, of course it will work. Neither of those things. So I'm going to nail my colors to the mast now. I think after looking at the HFEA data and the new data, I am convinced that if we were to perform a well-designed, well-performed RCT, it would undoubtedly show a clear benefit of PGT-A for advanced maternal age. Yes, we do know that study has to be performed, okay, and I fully accept that it does need to be performed. We do need to go ahead with it, but I'm in no doubt of what it would show, okay. Then you, the clinicians and the clinics individually, then need to make their clinical decisions based on that, I believe. If you want to read a little bit more about some of the things that we've done in this, please do refer to some of the papers we've published, also including our book that Gary Harton and I have released just in 2020. That has 3 chapters on this particular issue. And with that, I thank you very much for your time. I hope you've enjoyed the talk. It's been lovely talking to you.